Hello, good afternoon. Good afternoon, Bindu. How are you? 3 o'clock. Yeah, let's start the class. Right. Okay, good afternoon. Again, we are starting or continuing the biotechnology chapter. Right. Before that, please check am I audible properly and is the screen is visible properly. If you are having any issues, check your internet connection, also your device. If you still think that your internet connection is good, but still you're not able to hear me properly, just let me know. Okay, I will try it. If there is any problem from my end, I will rectify it. Okay. If you can't, if any of your friends couldn't join to the class, please tell them to put a message in the WhatsApp or Telegram group. One of our associates will help you out. Okay. So we'll start the class without wasting any time. We are discussing about biotechnology, second chapter, right? We already learned about the different aspects of biotechnology. Today we are like continuing that particular part. Okay. So as usual, before starting the class, we have to solve some of the question recall test, right? First recall test is coming. Yes, in a second. Sorry. Yeah. First question key. The question is simple, guys. Question what we discussed. Okay, the topic we discussed only. The question is the genetically modified or GM brinjal in India has been developed for. If you remember properly, 5-6 years ago, more than that, I think so. Okay, so there is a big chaos, like a lot of productions happening about the BT brinjal, which is developed in India, and a lot of farmers and a lot of politicians are against it. Okay, they're asking this particular genetically modified brinjal is developed for what they're asking. Option A is insect resistance. Option D is enhancing shelf life. Option C is enhancing mineral content, and option D is drought resistance. You have 30 seconds to answer. I'm expecting you guys to answer as quick as possible. Yes. We'll see who will give the correct answer. Okay. If you want one, what is that hint? Okay, so this brinjal name is BT brinjal. Okay, this is the hint. If you don't know after this hint also, it will be like BT brinjal. Okay, the name is BT brinjal. Okay, then I think you got the answer right. Bindu, what is your answer? Hello, for the new people joined. Yes. Okay. Okay. Your 1 minute is over rather than 30 seconds. I gave the 1 minute of time. Correct. So when we talk about BT means what? Bacillus thuringiensis. And what happens? It's have a crygene. What this crygenes will do? These crygenes will produce a particular kind of a toxins which are actually affect the insects, right? So BT brinjal means similar, right? It's also similar only. So BT brinjal is developed for the insect resistance. So option A is the correct answer. Remember, this question is asked in 2010. Okay, similar kind of questions maybe also asked this year. Okay, shall I move to the next question? If you have any doubt, let me know in the chat box. So I will answer that. Okay, second question. Always remember, if the question looks bigger, that means it's easier. Okay, don't think that when the question is big, oh, this question may be tough. No, whenever they ask paragraph kind of question or a question with like very long options and all, then the question is super easy. Okay, just I will read out this question, you will understand that. Okay, Bacillus thuringiensis forms a protein crystals which contain insecticidal protein. Okay, they're giving this data. Okay, there's a BT bacteria is there which are actually producing a protein crystal which contain an insecticidal protein, and this protein they're asking you have to fill it. Okay, option A binds with epithelial cells of the midgut of the insect pest, ultimately killing it. Okay, it's attached to epithelial cell and it will kill it. Okay, option B, it's coded by several genes including the gene cry. Okay, they're asking there are several genes are there, something there are like different name genes are there. One of the gene is cry. Okay, third option is activated by the acid pH of the foregut of the insect. First, fourth option is does not kill the carrier bacterium, which is itself resistant to toxin. They're asking it do not kill the what is that carrier bacterium, so it is self-resistant to toxin. Now you have to choose. So again, 30 seconds is there. Right answer, quick. Okay. Bacillus thuringiensis forms protein crystals which contain insecticidal protein. This protein, they're asking, okay, this protein does not kill the carrier bacterium, which is itself resistant to this toxin. Is it correct? Okay. What about the like our Bacillus thuringiensis is there? Is it resistance or what? No, this is not about resistivity of the bacteria. This is about which pH it works. Okay. So remember, this particular toxin works in the pH of alkaline nature. Right. So in the alkaline nature, it acts as a what is that? A toxin. Otherwise, it won't act as a toxin. Okay. Next, is activated by the acid pH? No, this is not activated by acid pH. It's activated by the alkaline pH. Is coded by several genes? No, it is coded by gene cry. In the cry gene, there are cry 1A, 2A, all those things. Okay. Binds with epithelial cells of the midgut of the insect pest, ultimately killing it. Okay, what happens? It will bind with the epithelial cells of the midgut of the insect pest, ultimately killing it. That's correct, right? What happens? It will attach to epithelial cells and it will perforate, or it will make a hole. And what happens? It will kill the insect pest, right? So A option is correct. So in the last class, what we did? In the last class, we are discussing about biotechnology used in agriculture. Majorly, we studied two things. The first one is a Bt cotton, and the second one we studied is our tobacco plant. There we studied about RNA interference, right? So next, our part we are studying is the biotech in the medicines. Okay, how the medicines it will work. Okay, so that's the second part. So, what we are discussing about biotech in medicines, like how we are utilizing in medicine. Before that, I just wanted to give us some idea about what it is called in there. Okay, the medicines, what we use the biotechnology, right? This is called as the red biotechnology. Red biotechnology. Red biotechnology means we are utilizing biotechnological process in either curing the disease or identifying the disease. Okay, those are the major things about the biotechnology in the medicine. Then why we use it? Okay, we use it for a different purpose. So the first purpose may be production of chemical or production of enzymes, production of the particular kind of the hormone, or production of the different types of the proteins. Okay, that's the major thing. And second thing, we normally use it for the gene therapy. Okay, we'll go in detail about what is gene therapy. Okay, third thing, we majorly use biotechnology in different aspects of diagnosis. Okay, we'll also see what's the diagnosis later. Okay, what's the meaning of diagnosis actually, and how we do it, those kind of things. Okay, and another one major things why we use the biotechnology is creating the different types of the medications or the medicines. Okay, different types of the medicines. So that's also one of the important thing. It may be vaccine or it may be regular drugs. Okay, everything is included in their only way. We can say like combiningly, we can say that. Okay, in a combining way, we can mention it like that. Okay, so those are the major aspects how we use the biotechnology in this particular medicinal field. The first one or the major one, which is for your study, is the insulin production. Okay, before we go for the insulin production, we'll go for like how insulin actually produced in our body or why we need insulin. Okay, I think most of you heard about insulin, right? What happens if the insulin production stop in our body? Can anybody tell me what happens? As I already mentioned, for the people who are joining from the school, keep one of your classmate or the class leader in front of the laptop or the projector, so he can give the answer in behalf of all of you. Okay, correct. So what happens when the insulin production stops or decreases? It creates a problem. That means what insulin actually do? Okay, insulin actually do is it will convert glucose into glycogen. Okay, glycogen. Okay, so what happens? You eat some sugar, something. Okay, everything is converted into glucose. Then what happens? Glucose will start circulating in your body. Remember, glucose in the blood is dangerous. Why it is dangerous? Because glucose can form the crystals, right? Think about, you are dissolving the sugar in the water. Okay, think about it. Just you add one spoon of sugar in the water, nothing happened. 2 spoon, nothing happened. Think about, like, you increased. Okay, keep on increasing, adding the sugar. Then what happens? It won't dissolve. Then you little boil it. After that, it will start forming the crystals, right? Think about this glucose forming crystals in our capillaries. What happens when it happens? Our blood vessels will destroy. Okay, blood vessels get destroyed. So our body wants to reduce it. How it reduces it? This particular, it produces insulin. This insulin production happens in pancreas. Okay, it happens in the pancreas. Okay, if you ask, so there are, in a pancreas, we normally divide into two different fields. Okay, one is the islets of Langerhans. Okay, another one is the synthesizing part. Okay, so in the parts of islets of Langerhans, okay, islets of Langerhans, there are three major types of cells you can see. Okay, one is alpha cells, another one is beta cells, another one is gamma cells. Okay, majorly in the beta cells. Okay, in the beta cells of the pancreas. Okay, beta cells of islets of Langerhans of the pancreas. Okay, I think you remember, right? Pancreas will be look like this, right? And here will be islets of Langerhans. In that there will be some beta cells. Okay, beta cells. So this is the pancreas. Pancreas you studied in the 11th class, right? Here's the islets of Langerhans. Okay, it's like a patches. Okay, islets of Langerhans are like a patches. So their insulin production happens. These insulins directly produced into our body. Then there what happens? It's a hormone, right? It will convert glucose to the glycogen. Glycogen can be stored, and our glucose level will decrease. Okay, but as soon as the human race is evolving, especially in the 1800, we start seeing people, okay, who are normally what we can say obese, okay, and also who is eating a lot of different types of the food. Okay, we started seeing those are suffering from a disease where what happens, their urine contain glucose or ketone bodies. Okay, we identified that particular disease as diabetes mellitus. Diabetes mellitus. Remember, you have to remember diabetes mellitus because there is another disease called diabetes insipidus. Okay, diabetes insipidus is a different disease which is related to kidney function. Diabetes mellitus is the one which is related to the sugar and related to insulin. Okay, so some people started getting this particular disease. Okay, this causing a lot of harm. Some of them are dying of the disease. Okay, then we discovered it's because of the insulin. Now we have to, their body is not producing the insulin. Then how can we do? We can provide it from the outside. But how can we provide from the outside? In this particular case, our first discovery of the insulin, or the first therapy of the insulin, happened in 1921. Here there are two scientists called Banting and Best. Okay, along with one more guy called MacLeod. Okay, what they did is they synthesized the insulin from the pancreas of dog and pig. Okay, they slaughtered some pig and dog. They took the pancreas. They extracted the insulin. Okay, and they given this particular insulin to the patients who are suffering from the diabetes mellitus. For their surprise, what happens? Their diabetes is started to cure. Okay, but there is a risk along with that. The issue is like we are extracting the insulin from where? Insulin from the dog and pig. Do you think it's completely comfortable to our body? No, right? So when we given the insulin, those patients who receive the insulin start developing a lot of allergic reactions. Okay, their disease is decreasing. At the same time, some of these allergic reactions are so high, some of them are dying. Okay, so that kind of allergic reactions also can be seen. So then what we did, we can't slaughter the human to extract the insulin. That's common, right? Then we studied how the insulin produced in our body then, because we thought of producing it. When they started working on that, they come to know that what happens is like in our body, insulin produced in a long chain. Okay, as you can see here, okay, it produced in a long chain. Sorry, I'll just delete this particular one. Okay, a long chain we can say like this A chain, C chain, and D chain. A long chain it will form. Okay, as you can see that the A chain have a 30 amino acids. Okay, remember we are talking about this particular insulin. Okay, insulin is a hormone which is a protein hormone. Okay, it's made up of the amino acid. So A chain contain 30 amino acids. A chain contain 30 amino acid. AA means amino acid, just remember that. Okay. And this particular C chain, guess how much it is contained? It contains the total 63 minus 30. How much? Correct. C chain contain total of 33 amino acid. And there's one more chain, B chain, which is contain 21 amino acid. Okay, don't confuse. Remember, here to here they given us a 20, and this one we say 21 amino acid. Okay, so they discovered that it produced or insulin synthesis happens in this particular manner. First A chain, next C chain, next B chain. Okay, I will explain why not B here, why not C here. Okay, that I will explain later. Okay, remember all these are made up of the amino acid. Totally how many amino acids is made up of? 30. So 60 plus 70, 80, 84 amino acid long, this particular insulin. And this form is called as a proinsulin. Okay, proinsulin. What's the meaning of proinsulin? Whenever you discussing about enzymes or hormones, if you heard the term pro, okay, that does not mean it's superior. Pro means it's not active. Okay, this pro insulin is actually an inactive form of the insulin. Inactive. That means it's not an active one. Okay, it can't convert glucose into glycogen. Its production happens in this particular manner. Okay. Later, this proinsulin in our body convert into insulin or mature insulin by losing the C chain. That means the mature insulin contain only A chain and B chain. Okay, only A and B chains are present in the mature insulin. Okay, understood, right? So let me explain once again. In our body, the insulin production happens in a long chain which contain A chain, B chain, and C chain. Okay, this is called proinsulin. That is the inactive form of the insulin. Then this inactive formed insulin lose its C chain and form the mature insulin, which only have A chain and B chain. Okay, so this A chain and B chain is connected by disulfide bridges. Okay, these are called disulfide bridges. Okay, disulfide bridges. So let me ask you a question, or let us think about one particular question. Okay, this answer is not even in the textbook, but regularly they may ask. The question is, because insulin made up of the amino acid, right? Insulin is made up of the amino acid, everyone of us know. And you can see disulfide bonds also present in between. So, which amino acid is responsible for the disulfide bond? Which amino acid is responsible for the disulfide bond or bridges in the insulin? Can you guys answer? You guys studied biomolecule in 11th, biomolecule in the 12th. Okay. And in molecular biology also you studied about this amino acid. Can you guys tell me the answer? Yes. Anyone? What you have to think now is, when I ask you which amino acids contain or which amino acid is responsible for the disulfide bridge, you have to think about two amino acids. One is cysteine, another one is the methionine. Only these two amino acids have the thiol group. That means sulfur group, right? But if you remember the structure which is given in the chemistry biomolecule chapter, okay, methionine has sulfur, but below that there is a methyl group is there. Okay, that means the thiol group is not open. There's a methyl group is there, so it cannot react. Okay, so then what left with? Remember, whenever you see disulfide bond in the living system or in the protein, that is because of or responsible of is cysteine amino acid. Wherever cysteine amino acid is there, the disulfide bridge will happen. Okay, and you also have to remember what is the number of disulfide bridges in the insulin. As you can see here, 1 here, 2 here, okay, and 3 here. Total 3 disulfide bridges will be present in the amino acid. Insulin. Okay, understood? Any doubt? Okay, now we got it. This is the production. Later, that is the issue what we caused. Insulin produced in a long chain, then it's converted into 2 chain with a disulfide bridge. That is what created the production of insulin difficult. Okay, then it's like a very long time. We don't know exactly how to prepare it. There are a lot of systems are there, a lot of technology we are using, but we are not producing a proper insulin. Okay, there our biotechnology came in. So what they did here is like they identify the gene which is responsible for production of the A link, okay, A chain, okay, and they also discovered the gene which is responsible for the B chain. And what they did is like they integrated this A chain in a plasmid and B chain in a plasmid. They inserted this plasmid in a E. coli bacteria and they developed it. That means what they are doing now, they are producing A chain separately, B chain separately. Okay, both of the A chain and B chain, what is that? Preparation or the formation happens separately. If we have all this data, okay. Now what we did, we actually joined these two. Okay, when we bring both of this chain near, what happened? Disulfide bridge happened. Okay, disulfide bridge happened. So that means we got the active insulin. Understood, right? What we did, instead of producing complete ABC chain and cutting the C chain, we produced A chain separately, B chain separately, and when we got the A chain and B chain, then we joined it, or when we bring it near, it formed a disulfide bridge. Okay, this particular discovery happened with the American pharmaceutical company called Eli Lilly. Okay, Eli Lilly. So this is the company which produced a proper active human insulin. Okay, they called it as the Humulin. Okay, Humulin. As you can see on this particular bottle. Okay, Humulin means what? Human insulin. Okay, first proper human insulin, that's called as the Humulin. Okay, just have to remember this is how we produce the human insulin. Got it? Any doubt? Simple, right? Now we'll see a question then, how the questions will be asked. The question is simple. Which of the following statement is not correct? Whenever this question comes, this kind, you just have to remember, you have to choose which are not correct, right? In man, insulin is synthesized as a proinsulin. The proinsulin has an extra peptide called C-peptide. The functional insulin has A and B chains linked together by hydrogen bonds. Genetically engineered insulin is produced in E. coli. Okay, I'm giving you 30 seconds of time. You guys have to answer as soon as possible. I'm just waiting. Yes. What about the others? YouTube. Okay, yeah, let's see the answer, correct answer. Okay, the first is saying in man, insulin synthesized as a proinsulin, right or wrong? The statement is wrong, but we need a not correct answer, so this is not the answer, right? The statement is correct. The proinsulin has an extra peptide called a C-peptide, right or wrong? The statement is correct, right? Because proinsulin have a 3 segments. One is the C-peptide, right? C-peptide. So this statement is correct. So this is not the correct answer. The functional insulin have A and B chain linked together by a hydrogen bond, right or wrong? This is the A chain, B chain. Is it a hydrogen bond? Is it hydrogen bond? No, right? Which bond it is? These are disulfide bond, right? Disulfide bond. So this particular statement is correct. So, sorry, this particular statement is wrong. That's why this answer is correct. The correct answer is option C. But still, we'll see the option D. Genetically engineered insulin is produced in E. coli. Okay, genetically engineered insulin is produced in E. coli. This statement is also correct. So this is not the correct answer as we studied, right? So we insert it in the E. coli. From there only we will get our insulin. If you have any doubt, please let me know before we are going to the next topic. Okay. Any doubt? Anything? You can just put that in the chat box. Okay, we'll go to the second important medicinal application of the gene therapy. Okay, so medicinal application of the biotechnology, that is called the gene therapy. Okay, if you ask me what is gene therapy means replacing or replacement of faulty genes to active genes or active or proper genes. Okay, think about, there are a lot of scenarios are there. Think about color blindness. Okay, we studied the color blindness. What happens because of a gene, because of the mutation in a gene, what happens, that particular person can't see all the colors, right? He maybe can't see red color or blue color, right? So based on that we have a different category. So in that, what is responsible? A faulty gene is responsible, a mutated gene is responsible. So if I remove that gene and if I put a proper gene, then what happens? He will get cured, right? So this is called as the gene therapy. Okay, we first use the gene therapy for our patients. Okay, a 4-year-old patient in the 1990. Okay, in the 1990, who is suffering from a disease called ADA deficiency, or in a full form adenosine deaminase deficiency. What happens with the people who is suffering from the ADA deficiency is adenosine deaminase is very important in maintaining our immune system. Okay, when this particular deficiency happens, usually because of an enzyme which convert this. Okay, so that particular patient or that particular person do not have that much of immunity, so he's susceptible for all the basic level of a disease. Okay, even a common cold can kill that person because you don't have the immunity at all, right? So a 4-year girl now had this particular condition. So what we did is, as you can see the picture, okay, we took the cells from her. This adenosine deaminase enzymes will be produced from the WBC cells. Okay, what we did is like we removed the cells from that particular patient, from the girl. Okay, at the same time, we in the laboratory, we created the one virus along with our desired DNA. Desired DNA means a proper DNA or active gene. Okay, and in the lab only, we made alteration to that particular cell. Okay, alteration to that particular cell. And when the alteration happened, we inserted the same cells into the body of that particular person or that particular patient. Now what happens when we are taking out, it have a faulty genes, right? When we are inserting again, it have a working genes. So once it's inserted again in the body, this genes functions normally, and this particular person will get the ADA, okay, ADA enzyme, and this immunity will again back on. Okay, but there is a question: does this gene therapy is permanent? No, it is not permanent. If you take a RBC, you can't do anything in the RBC because mature RBC do not have the chromosomes. Okay, think about WBC. If you take a WBC, if you made the alteration, but it can live for some days only, some months only. It can't live for like throughout your life. It will die, right? What you have to do? You have to do it again and again. This is good for one thing. You have to do it again and again. And the second thing, can you change the genes of all the cells in our body? Can we do that? No, that is not possible, right? You can do it for like slight kind of thing, but you can't do it for the complete body. That's not possible. Our body contains millions of billions of cells, right? You can't do that. Then how can we do it? We can do this if we do the alteration in the zygote. You know, right? What happens in the zygote? The zygote is a single cell. Zygote is a single cell, right? If you make any changes in the zygote, okay, if you made any changes in the zygote, what happens? That zygote develop into complete body. So whatever the alteration you do in that particular single cell can be distributed to throughout the body. Okay. Then why are we not doing it, right? This is a simple task. We just have to take a zygote and we have to do it and we have to grow it, right? It's so super simple, right? Some of these things will come called ethical issues. Okay, so sometimes it's not like it is not possible. Okay, sometimes it's most about the ethical problem. What is ethical? Okay, ethical means which are good to the society, which are bad to the society. Okay, because of the ethical issues, we restricted certain things in the biotechnology. Okay, why we restricted? One of that restriction is modification of genes in the humans. Okay, modification of the genes in the humans. Okay, so there are two reasons why it is. One is it's a difficult task. It is not simple as it seems. Okay, you have to take several set of zygote, and in that one or two may get successful. That means the success rate is very low. Okay, that's the first case. Second case is majorly about, think about, there are terrorists. Okay, they can create, or there are people who are like mad. Okay, what they can do, they can start using that gene, this gene and all, right? They can start working on different genes and they can start creating all the wild different kind of the organisms. That's the second one. And there are a similar kind of a lot of ethical issues that we'll discuss one more time. Okay, so because of that we have restrictions for that. Okay, and also because it have a very low chance of success. Okay, so as I mentioned, in the adult body you can't change, you can't apply gene therapy to every cell because we have billions of cells. Okay, but if you make changes in the zygote, which can be carried out, but we are not in that particular level of this one. Okay, so that's why it's not reached that particular level. For you guys, you just have to remember it's like gene therapy we actually implemented for the adenosine diphosphate for a girl. Okay, a 4-year-old girl. Okay, and majorly it's also can be used for the anemia and also SCID, severe combined immunodeficiency, and also for the our anemic disease. Okay, so those are the major implementation of the gene therapy. Okay, if you have any doubt, let me know. So next we'll go for the molecular diagnosis, how we use biotechnology in a molecular diagnosis. Let me put it in a very simple term. Okay, so biotechnology we studied about PCR, right? So what is the diagnosis then? Diagnosis means identifying the disease based on the symptoms. Okay, based on the symptoms. But there's a chance for that. Okay, because when the symptoms are start to form, when diseases normally spread to our body, think about like corona. Okay, think about today one person got the corona. Do that person start showing when he got the corona? Think about like corona is here. Okay, it come and like enter into my body. Suddenly I will start showing symptoms. No, right? The symptoms will be start showing after 2 or 3 days. Right, until that the person will be normal only. But in the normal time also what he can do? He can spread to the other, right? He can spread to the other, and at the same time what's happening? His body, inside his body, the virus is attacking our body system and all. Understood? So diagnosis issue is, you can use the diagnosis, but when the symptom arises, the diagnosis is based on symptoms. So that is why we developed another level using the biotechnology called molecular diagnosis. Okay, molecular diagnosis means here we don't need symptoms. We majorly work based on the chemical constituents. Okay, we majorly work based on the chemical constituents. The very simple example is RT-PCR. Okay, very simple is the RT-PCR. So as we seen, I also explained what is RT-PCR in the last class, right? How we do the RT-PCR. So think about something there. Okay, think about there's a one person. He's very normal. Okay, he's like he do not have any symptoms and all. Okay, so but he have a doubt that he may have a corona because he traveled in a bus. Okay, then what he can do? If he go to doctor can't say that he have the corona or not because he do not have any symptoms. But if you took the RT-PCR test, if the result come positive, that means he have a corona, even though he do not have the symptoms. Understood? Molecular diagnosis help us identify disease earlier than the onset of the symptoms. Onset of symptom means when the symptom comes. Molecular diagnosis help us to identify the disease before the onset of the disease. Okay, we can also use molecular diagnosis. COVID-19, okay, SARS-CoV-2 or COVID-19, and we also use this for different diseases, something like AIDS. Okay, also for the hepatitis B, all different types of disease. Okay, there is, when we are doing this molecular diagnosis, what we do is like we'll take either part of the DNA or part of the RNA or part of the protein for running along the our library. Okay, it's very simple. Let me put it in this way. Okay, so think about I gave you a book. Okay, think about I gave you a book. Okay, but that book is sealed. Only you can see the front. Okay, only you can see the front. You want to understand how the book is, right? And I will leave you in a library. What you will do, you will try to recognize similar kind of the book anywhere in the library. If you find the book, what you will do, you will open and read it and you will understand what's in the book I given to you, right? Same. In the molecular diagnosis, we have certain radio labeled means we put some radioactive molecule, okay, radioactive molecule either to DNA or to RNA, okay, and we'll leave it along with our samples. In the sample, if the virus is present, this our radio labeled probe go and attach here. Okay, radio labeled probe go and attach here, identifying that the virus is there. Okay, that's how RT-PCR work. They will take your nasal sample, right? Nasal and this pharynx samples. Okay, nasopharynx and osopharynx samples, right? So what they do, they run in a PCR machine. There, what happens? If you have a virus, it will show a color or it will show graph. Okay, because our probe going and attaching to it. Okay, that's simple it is. So same you can also use it in the AIDS also. Okay, so majorly if you use DNA to doing this diagnosis or to run this, okay, this is called as the southern blotting. Remember, it's based on a scientist name. DNA means if they are running DNA or separation of DNA, it's called southern blotting. Always remember. Okay, southern blotting. Then what about RNA? Exactly opposite. If it is RNA we are separating or identifying, then it's northern blotting. DNA southern blotting, RNA is northern blotting. What about protein then? If I use the protein, then how it called? It's called as the western blotting. Okay, western blotting. Don't ask me, sir, where is eastern blotting? There is nothing called as eastern blotting. Okay, there is nothing called as eastern blotting. Only three types. Southern blotting means you are recognizing the DNA or separating the DNA. Northern blotting means you are separating or identifying RNA. And western blotting means you are separating or identifying the protein. As simple as it is. Okay. There is another technique, PCR which majorly works on the basis of DNA or RNA. Okay, majorly DNA. And this is another technique we also used called ELISA. Okay, ELISA means, I think you heard about it, this is the diagnosing method for the AIDS, right? This is an antigen antibody reaction. Antigen antibody reaction. There also we developed a special kind of the antibodies for special kind of antigen. Best example again COVID. There is another test called RAT test, right? Rapid antigen test. What they will do in a rapid antigen test? In that they will take a kit, they will put some drops of your nasal drop or something. Then if the mark or 2 mark comes, then it's positive. A single mark comes, it's a negative. Right here, these lines represent each of these lines. Okay, one line will contain antigen. Okay, one line will contain antigen. Sorry, antibody. One line will contain antibody, and another one line is called reference line. Okay, reference line. Think about you put your samples here. These samples will be start moving here, right, in this direction. What happens is like, if this particular sample contain the antigen, then what happens? Antigen antibody reaction happens and it will show the first line. Okay, it will show the first line. And when it is moving, this reference line will use to check that that machine is working or not. Okay. If this line also appear, that means machine is working. If this line is working, that means shown, that means antigen is present in the sample. That means that is positive. Okay. If it is only one line is coming, means what? This machine is properly working, but there is no antigen. That's called a negative. So same reaction in an advanced level we'll do called ELISA. ELISA means enzyme linked immunosorbent assay. For the student, I'm telling you one more time. Remember, please write it down if you never heard about it. ELISA means enzyme linked immunosorbent assay. The questions will be asked about the detail. So molecular diagnosis, if I want to inform you in a nutshell, what is molecular diagnosis? Help us to recognize the disease before the onset of the symptoms. Okay, majorly I can divide into PCR and antigen antibody reactions. PCR majorly works with the DNA and RNA, and our ELISA majorly works with the antigen antibody reactions. Okay, molecular diagnosis can be used with the DNA, RNA, or the proteins. DNA, if you are using separation is happening, it's called southern blotting. If you are using RNA, it's called northern blotting. If you are using protein along with the antibody, it's called as the western blotting. Okay, details are not needed for you guys now, so no need to take tension. Okay, if you have any doubt, let me know. Okay, shall I continue? Okay, one question came. The first clinical gene therapy was given for treating. The first clinical gene therapy was given for treating. The first option is diabetes mellitus. Second option is chickenpox. Third option is rheumatoid arthritis. Fourth option is adenosine deaminase deficiency. Yes, for the people who don't know, rheumatoid arthritis means arthritis, you know, right? Joint pain. Okay, there is a different types of the arthritis are there. One of the type is rheumatoid arthritis. It's like autoimmune disease. That means our body only create that problem, the joint pain. Okay, in the aged people, that's rheumatoid arthritis. Okay. Okay, give me your answer in the chat box. I'm just waiting either in YouTube or in the Zoom. Anywhere is fine. Time is over, guys. We'll see. As I mentioned, the first clinical gene therapy happened for which particular disease? Adenosine deaminase deficiency happened for the adenosine deaminase deficiency. This question is asked in 2012. Okay, shall we move on? Next, we'll use what is the use of transgenic animals? Like, what is transgenic animals? We already seen. We made some alteration in the genes. Why we need to use this, right? What is the function or what is the use of this? These are the major 5 reasons why we use our transgenic animals. The first one is the normal physiology and development to recognize. Think about like we have like hundreds of the genes, but we don't know which particular genes is responsible for which particular one, right? So in that particular, what we will do, we will take a mouse, okay, we'll take a mice, we'll remove one gene, okay, we'll remove one specific gene, okay, one specific gene is, we remove it and we'll let it to develop, okay, we'll let it develop. Yeah, and we'll observe how the developmental pattern happens, what are the problems it will achieve, okay, and whatever, think about something, you removed one gene and you started growing that organism, and you can see that its brain development is not proper. That means what? That means this particular gene is responsible for the development of the brain. Okay. Think about you removed another gene and you start developing that mice and you recognize that that rat do not have the tail. That means what? That gene is responsible for the tail. So to identify the normal physiology and development, we'll use the transgenic animals. Okay. Next one is the study of the disease. So we normally, whatever the disease comes, we have to understand how the disease works within the laboratory condition, okay, to make us or to find the vaccine or the medications, okay. So you can't do the experiment with the humans, right? You can't like take all the patients and you do the experiments because human life worth a lot, right? But we can do the same thing with the mice, or we can do the same thing with the other transgenic animals. So what we do, we'll create the mice, transgenic mice, and we will infect the disease to that particular thing about tuberculosis. Okay, we infect that particular mice with the tuberculosis and we'll study how that particular transgenic mice works or they behaves. Okay. So what we normally do is like we use a sensitive kind of the animals, okay, and we will do the study to understand how the TB spreads, how it affects, how to make the changes in the body. Okay. Third one is a biological product. As we mentioned, you can get the biological products from the animal. Think about like animal E. coli, right? Similar to E. coli, you can create plants with higher abilities, okay, with very good abilities, or a product something like hormone producing banana. Okay, think about it. You identified a gene and you replaced in this one in the banana. So banana is producing hormone now. Instead of taking an injection, what you can do? You can eat a banana and you will get the hormone supplement. Okay, you can do that kind of things also. Okay. Next one is a vaccine safety. Before, don't think about whatever the vaccine is created. Think about like Covaxin, Covishield, whatever it is, right? COVID. We are not given you directly. Once we develop, we normally try this on the animals, okay, and we will try to identify what is this reactions or the problems which is causing, and we'll make the changes. And again, we kept it on the animals. When animals, it do not cause any reactions, okay, it do not cause any secondary reaction, then we'll try it on the humans. Okay, so for the safety of what is that vaccines also, we normally use the transgenic animals. Next one is a chemical safety testing. Okay, there are a lot of chemicals which we'll be using and all. Okay, but the issue is like we have to see that how this chemical affects to our body. So here is also we will use sensitive animals. Okay, sensitive animals. These sensitive animals means a regular animal. Think about like a regular animal show the reaction at 1 gram of the chemical. Okay, a regular animal show symptoms at 1 gram of the chemical, but this sensitive animals show the reaction at 0.001 or 0.01% or 0.01 gram of solution only, it will show the reaction. That means even a tiny amount also show the reaction. Think about you are adding 1 gram up to that particular animal. Then what happens? The symptoms will be in a very high level. So you can identify the symptoms easily. You can identify the problems easily. Okay. So to identify the problems or to identify the safety, we will also use the transgenic animals. So these are the major 5 reasons why we use the transgenic animals. Okay. If you have any doubt, please ask me. So when we are doing about transgenic animals and transgenic species, okay, there are certain valuable mention I want to do. First one is Rosie. Okay, Rosie is a cattle. Okay, we developed cattle. We developed for the protein enriched milk, to get the protein enriched milk. Protein enriched milk, it have almost like 2.4 grams of the protein per liter of the milk. Okay, and we also created transgenic mice regularly because of the production of different types of the what is it, sensitivity, all this kind of things. Okay, and we also created a modified transgenic dog called Doggie. Okay, DOGIE, which have the high sensitivity or excellent smelling capacity, smelling power. Okay, the first genetically obtained food product or genetically obtained food, first genetically engineered animal for the food is the salmon. Okay, we made some changes in the salmon for the food production. This is the first food production, first transgenic animal which we created for the food production. Okay, and we also use this for the different types of the other animals to increase their weight and increase the abilities. Okay, and we also created the transgenic sheep. Okay, transgenic sheep, which for the better growth. Okay, so these are some of the valuable mention. You just have to remember the Rosie, not others are not that much important. Okay, so when we'll go for the ethical issues. Ethical issues contain, there are a lot of things. Okay, that means, as I mentioned, which one is not good or which one is bad, those kind of things. Okay, here we are not going to the in-depth of the things. Okay, that's not important. We'll see a lot of one to two things. Ethical issues means which one is good to the society and which one is bad to the society. Okay, just have to determine that. To regarding that, we have a different committees. So before you are going to test anything on the animals or anything on the humans, you have to take the permission from the ethical issue committee. Okay, we have to take a permission. Otherwise, what happens? Everyone start experimenting with all the animals. That's really dangerous, right? So ethical issues are very important because we are living in a society, right? So along with the ethical issues, another very important thing happens is biopiracy. Okay, piracy means, like stealing something. Biopiracy means stealing something which is biological important. Okay, so I will give an example later on, but think about something. I spent my 10-15 years of life to create a particular kind of a medicine. Okay, I created it. But next day, everyone is started faking out. Everyone is like started taking that and started producing it. Then what happens? Whatever I spent in 10-15 years, everything gone into waste, right? So because to protect the producer or that person who created the things, we developed two major things. One is the patent and another one is copyright. Okay. Patent will be given to the person who invented something or the inventions. Okay, inventions. Think about Eli Lilly got the patent for the insulin production. Okay, if you create something or if you invent something, okay, you will given the patent. This patent normally valid for the 10 years. Okay, after the 10 in this particular 10 years, nobody can copy whatever you created or invented. Okay, nobody can copy whatever you invented. Okay, what happens if anyone want to make a similar product, they have to give you some amount of money. Okay, so some amount of money and take your permission, then they can do it. Okay, copyright is for anything which is documented or art related, I can say. That means you made a procedures. Okay, you created a procedure for a biotechnology. Okay, or you written a book or a paintings. Okay, procedures, books, literatures, or the painting, these are given the copyright. Okay, nobody can copy that without taking your permission. Copyright also works similar to the patent, only it has a certain validity. Okay, within the validity nobody can copy that. Okay, so after that you can extend it, or otherwise after that everyone can use it. Okay, so we came to, like, Indians came to the importance of these ethical issues majorly because of the some reasons. Okay, we'll see what are the two reasons. So whatever I'm mentioning, genetically engineering, what we are created in India, it have to gone through a committee called Genetic Engineering Approval Committee. Okay, Genetic Engineering Approval Committee. Okay, so as I mentioned, biopiracy is the something where someone is stealing your product. Okay, it happened to us several times. So the first or the best example is about taxmati. Okay, so everyone know about the basmati, right? Basmati rice. So basmati is an Indian rice which normally grown in the foothills of the Himalaya. Initially, now it's widespread in India, right? Initially, it's grown in the foothills of the Himalaya. It have a kind of aroma, right? The grains are normally longer and it have a kind of aroma. It's very famous. India export, there are thousands of tons of the what's that rice to the world. Okay, what happened in the 1996, I think, okay, 95, 96, that time, there's a company from the US called Rice INC from the America. Okay, what they did is like they took this basmati rice. Okay, they hybrided with a short or the dark variety, and they call it with a name called taxmati, and they put for the patent to that. Okay, so if they sell it, what are the two things happen? Because they are selling our basmati in their name. Okay, the export from India is reduced, right? Export from the India is reduced because nobody wants to import when they have their own food, right? Second thing, because they took a patent, okay, because they applied for the patent, Indians can't sell our basmati in the America because the patent is with the American company. Okay, we can't do it because we have to go and ask permission to that private corporation, right? So then we went to the international court regarding that. Okay, then international court asked for creating the new variety, but they failed to give the what is that all the documents. Then international court given the order towards us. That means they asked them to not sell taxmati in the America, okay, with the name, okay, and so patent is withdrawn, okay. After that, we start selling the basmati again. Another very important story here is about the name. It also happened in the 1997, I think. Okay, what happened is like some of the companies or the pharmaceutical companies of the Europe, they started taking the patents for the different neem products. Okay, neem product in the this one, I think everyone of you know neem, right? Having a bitter taste, right? That one. Okay. So they took the patent for its antibacterial property. They took the patent for its active, usage in the agriculture. Okay, different types of the patent they took. But the issue, what happens is like, even though in India, if I want to create something of the name, what happens? I have to ask the permission to a company which is present in the Europe. Okay. Then we again went into the international court regarding the same issue. Okay. There also again that particular came to us, like the order came in behalf of us saying that name we are using it from several hundred years. It's a traditional knowledge, right? It's a traditional knowledge. It cannot be patented because we are using it from the as antibacterial, as agriculture in a lot of hundreds of years. So this usage of these companies, American and European companies, without taking the permission is called biopiracy. Okay, biopiracy. Okay, so this is one of the important aspects in the biotechnology we should be aware of. Okay. So one question came, so last question, try to answer quickly. A new variety of rice was patented by a company. So through such varieties have been present in India for a long time. This is related to CO667, Sabarmati Sonora, Lermo Rosso, Basmati. This question is asked in NEET 2018. Can you guys answer? Can you guys answer? Okay. Correct. The correct answer is what? Correct answer is Basmati. Correct answer is Basmati. Okay. So that's the today's class, guys. Before you are going out, I have to inform you regarding two things. One is the test alert. Tomorrow you have a test. Biotechnology principle and process is the chapter. Okay, not the application, only principle and process. There will be 40 question will be there. You will be given 40 minutes for the every correct answer. You will be awarded +4 marks. Every wrong answer you will lose -1 marks. If you do not attend it, there will be no lose or no gaining of the marks. That will be 0. Okay. Please be well prepared. Tomorrow afternoon, the test will be there. Okay. Notice will be sent to your coordinator numbers. If you are in the home, it will be sent in the your WhatsApp group also. Okay. Before coming to the next class, please read the biotechnology chapter and also make sure that you are using the same SRN ID, student registration number, or the same mobile number. Do not use a different mobile number every day. Say use the same mobile number for every classes. Okay. So that is the class for today. If you have any doubt, you can ask me now. Okay. If you do not have any if you do not have any doubts, we can leave the meet. We'll meet again tomorrow morning. Okay. Okay, great. We'll meet tomorrow morning. Be prepared for